Okay, this slide presentation is on respiration for Leaving Cert Biology. This slide shows the syllabus requirements. We start looking at the definition and role of aerobic respiration. So respiration is the release of energy from food using oxygen. Why do living organisms need energy? They need energy to carry out their metabolic activity, the chemical reactions within their cells. So metabolism is the sum of all the chemical reactions that are happening inside an organism. We can look at two types of metabolism: anabolic reactions and catabolic reactions. Anabolic reactions, such as protein synthesis, need energy. They're going to build large molecules from smaller pieces, whereas catabolic reactions are going to break down larger molecules, and they often release energy. Aerobic respiration, for example, is a catabolic reaction. Now, the overall equation for respiration makes it look quite simple. We have two reactants and two products, but there's a long process involved in between. But if we're looking at the reactants, glucose and oxygen, it's very important that you know the formula for glucose, C6H12O6, and there's 6 oxygen atoms involved, so 6O2, and that produces 6 waste molecules of carbon dioxide and 6 waste molecules of water. When this happens, lots and lots of energy is released, and this energy is used to form molecules of ATP within the cell. We can look at aerobic respiration in 2 stages. Okay, in stage 1, it doesn't require oxygen and produces a very small amount of energy. Stage 1 of respiration happens in the cytosol of the cell. This is the cytoplasm without the organelles. So glucose is broken down to small molecules, releasing a small amount of energy. In stage 2, does require oxygen and releases a large amount of energy. And in stage 2, the molecule of glucose is completely broken down into smaller molecules of water and carbon dioxide. So stage 2 occurs in the mitochondria, needs oxygen, will produce a larger amount of energy. So we're going to look at the first stage in a little bit of detail. The first stage is called glycolysis. Glycolysis literally means the splitting of glucose molecules. So the glucose molecule is split in half into 2 smaller molecules. These smaller molecules have 3 carbons each, so they're known as 3-carbon compounds. They're called pyruvic acid. So glucose is split using enzymes into 2 molecules of 3-carbon pyruvic acid. Now, in that process, a small amount of energy is released, enough to form molecules of ATP. So ATP is this energy-rich molecule. It's formed by combining ADP and P. Okay, if there's no oxygen present, okay, it remains a stage 1 process. The pyruvic acid is going to be converted to either ethanol or lactic acid. It's going to be converted to lactic acid in muscle cells and in many bacteria, or in yeast cells, the pyruvic acid can be converted to ethanol with some carbon dioxide being released as well. Now, looking at the second stage, this is the stage where a lot of energy is being produced. In stage 2, the pyruvic acid moves into the mitochondria. So this will only happen if there's oxygen present. Once it moves into the mitochondria, it's broken down further into a smaller molecule called acetyl-CoA. It loses a carbon dioxide molecule at this stage. The acetyl-CoA molecule enters a series of reactions called Krebs cycle. Krebs cycle happens in the lumen or the center of the mitochondria. During Krebs cycle, the acetyl-CoA is completely broken down. Okay, the end products of this breakdown are carbon dioxide, protons, and electrons. The carbon dioxide is released. The protons are picked up by a proton carrier called NAD. So NADH is formed. NADH also picks up the electrons. So we have this carrier molecule called NAD, which picks up the protons and electrons and carries them to the electron transport chain of reactions, which is found on the cristae of the mitochondria. The cristae are the infoldings on the inner membrane of the mitochondria. Lots of enzymes are located there, and these enzymes are part of the electron transport system. Electrons pass along the transport chain, releasing energy to form ATP. So all of these energized electrons that are released from Krebs cycle, they all end up in the electron transport system on the cristae. They're energized. They pass along the carrier system and their energy is lost, and that energy is used to form ATP. Eventually, the electrons return to the proton and combine with oxygen to form water. Now, this is an overall summary of the process. Starting at the top, we have our glucose. The first stage, which happens in the cytoplasm, the glucose is broken down into pyruvic acid. Okay, that's called glycolysis. There's just one of the pyruvic acid molecules drawn here. There actually would be two. If this remains an anaerobic pathway, the pyruvic acid is going to be converted into ethanol and carbon dioxide in the case of yeast, but it will be converted into lactic acid in the case of a muscle cell or bacteria. If there is oxygen present, the pyruvic acid can enter the mitochondria, and here we can see it's broken down into acetyl-CoA. It loses the carbon dioxide. There's some protons and hydrogen released there. The acetyl-CoA enters Krebs cycle, and in Krebs cycle, we have the complete breakdown of acetyl-CoA. It's broken down into carbon dioxide, protons, and electrons. So in this diagram, the protons and electrons are collectively referred to as hydrogen. So the hydrogen, remember, which consists of protons and electrons, are picked up by NAD, forming NADH. Okay, carried to the electron transport system, where all of those energized electrons pass along the electron transport system, releasing energy to form ATP. Eventually, it's passed on to oxygen to form water. So the electrons and protons are passed on to oxygen to form water. Okay, another look at anaerobic respiration in detail. So anaerobic means without oxygen. It's the release of energy from food without oxygen. It's entirely a stage 1 process, so it entirely happens in the cytosol. There are two possibilities, depending on what type of cell it is. If it's in a yeast cell, the glucose is converted into alcohol and carbon dioxide, or the alcohol and ethanol, or ethanol and carbon dioxide. If it's in a muscle cell, for example, or many bacteria cells, the glucose can be converted to lactic acid. And in each case, a small amount of energy is released. Alcohol and carbon dioxide are the products of respiration by yeast cells, and those products are used in brewing and baking. Okay, the alcohol obviously used to make alcoholic drinks, and the carbon dioxide is used to make bread rise. Lactic acid is a product of anaerobic respiration in muscles. So lactic acid can cause pain and muscle cramps, and it needs to be transported to the liver to be broken down. Lactic acid produced by bacteria is what causes the pH of yogurt to drop in yogurt production. Now, another look at some of the industrial uses of fermentation. So a couple of key words. Bioprocessing is any process that uses complete living cells or their components. So an entire bacteria cell or a yeast cell or even a chloroplast or an enzyme, to obtain desired products such as vitamins, food additives, and medicine. So it's a process where cells are used to produce useful products. Some examples: yeast is an organism capable of anaerobic respiration, and we use yeast to ferment sugars and carbohydrates into alcohol products such as beer. Ethanol and carbon dioxide are made when yeast ferments glucose. Immobilized cells such as immobilized yeast can be trapped in sodium alginate. The yeast cells continue to ferment glucose but are easy to separate from the product. So I know the immobilization experiment is carried out when you study enzymes. It's just briefly mentioned here. An advantage of immobilizing cells within a bioreactor is to make them easier to reuse those cells or cell components, and it makes it easy to separate them from the product. So a bioreactor is a container in which you can control the conditions such as temperature and pH easily. So this is a practical or a mandatory experiment we have to do, is to prepare and show the production of alcohol by yeast. So there's two parts to this practical. First of all is preparing alcohol and then show its production at the end. So when preparing the alcohol, we're going to use yeast and glucose. Glucose provides the food for the yeast. We use boiled, cooled, water. So boiling water removes the oxygen. And we need to cool it back down again to a temperature suitable for the yeast. A control would be a second flask, the same apparatus without yeast. So the control flask would have the boiled water and the sugar, but wouldn't have the yeast. A fermentation lock or aerobic is used to keep out oxygen. Okay, and that ensures anaerobic condition. It allows the carbon dioxide produced to escape, but won't allow oxygen back in. It also used to keep out harmful bacteria that might contaminate the experiment. Carbon dioxide is produced during the experiment and can be seen as bubbles. Okay, we know that fermentation is finished when it stops bubbling. Depending on the type of yeast used, the bubbling could stop after a few hours or could stop after a day or two. Once the fermentation has stopped, filter the mixture into a test tube. So you now have a filtrate that contains alcohol. So we need to filter out the dead yeast cells. Remember, the yeast cells will die eventually because alcohol is toxic to them and will build up in the environment and kill them. So to test our filtrate to see does it contain alcohol, we use what's called the iodoform test. Add potassium iodide and then add sodium hypochlorite. Okay, so two chemicals, add potassium iodide and then add sodium hypochlorite and heat the solution. The color changes from orange to yellow, showing alcohol is present. So we get a yellow color at the end. So for that, we're remembering the two chemicals, potassium iodide and sodium hypochlorite, and the positive color at the end is yellow crystals.